So what do I do at Dana-Farber? Right now I'm the Chief Clinical Research Officer. My background is that I initially worked at the National Cancer Institute for about 17 years before I came to Dana-Farber. And there my role was to study the relationship between changes in the different gene copy numbers and mutations that Barrett talked about that we had the capability of doing. When I started doing in the 1980s, we could look at one gene at a time. It used to take me months to find out if there were multiple copies of genes that were present in lung cancer cells. And now that's dramatically changed. Another part of my job is to see and take care of patients with lung cancer. And lung cancer's had a bad reputation. The initial time that I got into it was that I thought it was very important to work on this disease, the most common killer of people with cancer, and also that these people needed some basic science to bring better treatment to their lives. My current job, that's an unbelievable pleasure, is to begin to see what's happening as we're applying the findings from science, not only discoveries, but the systematic characterization of the tumor, and bring some of more effective treatments to our patients with cancer. The other part that's dramatically changed is my very first study that I did and published in 1985, it's now 30 years ago, was reporting on 5-year survivors of a certain kind of lung cancer, small cell lung cancer, and reported on 5-year survivals, which at that period of time was very unusual, and it's one of the markers that we use as a potential for what we call cures in cancer. And I'm going to try to bring all that in. I want to make a couple of points going forward. Number one is that I want to show you how part of this has been accomplished by taking things that we thought were the same by looking at under the microscope even a decade ago and breaking them down into subsets that can make more effective treatments. And I'll show you a couple of examples. The second thing I want to get across is by taking a look at an enterprise effort. As we've heard in the themes before, we're much stronger together than we are working individually. And we're trying to take the lessons taught to us by our colleagues studying other types of cancer, pediatric cancer that you've heard, number one. Number two is to try to find out other tumors. I've worked in lung cancer all my life, but now I get to work more broadly across the Institute in my role as the Chief Clinical Research Officer. I'm going to give you 2 examples that took place starting a decade ago that began transforming our field. Could I have the slides, please? And I want to now the point here, if you take a look at the panel on the right and left, these are both adenocarcinomas of the lung. The reason why we picked these is that they're supposed to look relatively similar. Now, these are pictures if you take somebody's tumor out and you look at it under the microscope. These are called photomicrographs. And the purpose is they're supposed to look similar. These are made up, those pink cells are lung cancer cells. And the things that kind of go through there as striations are the blood vessels that go to it to supply it. Now, even though these look exactly the same under the microscope, when you take the DNA out that Barrett talked about, and you begin to sequence it, which when we initially did this was a very big challenge, and we used to also have to do this one gene at a time, and this was only a decade ago, is it turns out you have a genetic change that Barrett talked about. And it so happens, he gave the example of the skin and what normally happens with your skin if you cut it and you get signals that turn it on. And in this particular one, it turns out there's a gene, the EGFR stands for epidermal growth factor receptor. The normal function of this in your skin, the epidermis is your skin, is that if you injure yourself, i.e. you get a cut, it starts to regrow and heal itself. Now, it turns out for whatever reason or other, in adenocarcinoma of the lung, you get a genetic change in this particular gene, and you can end up giving a drug that specifically turns that off. Now, that discovery was made because we found things that Barrett talked about, and that is that there were people that had these exceptional responses that would get this drug, and it would go on, instead of for months, it would go on for a year or even longer. And so working with Matthew Meyerson from the Basic Science group, Passiani, and a group of others, we sequenced this and found out, lo and behold, there's a genetic change in there, and it turns out that's why it's very sensitive. Now, the problem is that this is present in only about 15% of lung cancers. And they all look the same, so you end up having to systematically genotype them. We thought this was transformative. We thought instead of taking a look and having chemotherapy only work a matter of months, you could get this and it would work for a year or years. So we thought this is very important. We need to know this about every one of our patients. And because of many of those of you sitting in the room, we could just begin doing it. We began systematically genotyping all of our patients starting in 2004. And as Barrett told you, we still struggle to get companies to pay for it. But we thought this was transformative, so we started doing it, and doing it consistently. By 2005, there was a commercial test, but the companies didn't begin paying for it. The insurance companies and the third-party payers didn't start paying for it until about 2009. So for 5 years, we consistently genotyped this for all of our patients. It was hundreds a year, and we did this. The other thing that happened was that the one on the right turned out to be a different change. And in this one, instead of having a genetic change, it was a break apart and rearrangement of things called chromosomes. It's the way our genes are packaged. Now, unfortunately, we didn't make this discovery. One of the things that happened, well, we can't discover everything. It was discovered in Japan in 2007. But because we knew about it, Passiani's lab and the lab from Mass General ended up testing existing drugs that had been developed for another person, for another purpose, and ended up finding out that you could treat cells that had this abnormality, and as Lauren talked about, you could actually make them die. It caused this cell death. And this is present in 5% of lung cancers. And you only have one or the other. And as Barrett talked, these are all drivers. So you began having to test 2 genes, and we started doing that in 2008. Now, the other part I wanted to get across is it's critically important not to learn just what you're learning from lung cancer, but to learn from what's going on in other tumors. And we decided that we needed to have systematic characterization of this starting in 2004, and we began studying 6 genes in 2008. We went to 10 in 2009, we went to 14 in 2012, and because of Barrett's work and the works of the Institute, we took the big jump 2 years ago and went to 300. And what we do is we systematically do this for all of our patients, and we've been doing it now for a decade, mostly supported by people here in the room of doing this because it's transformative. And I wanted to give you a second example of what's happened, and this is one of the things that I work on and run a trial across the world, actually, of looking at something called BRAF. Now, BRAF is a different gene. It was discovered in melanoma. About 2/3 of patients with melanomas have a genetic change called a BRAF mutation in a specific part. And companies had developed drugs that uniquely went after this specific change within this tumor. And they began treating melanoma patients about 5 years ago. Because we'd systematically characterized our patients for this, what we learned in melanoma, we began to apply in our patients with lung cancer. And by turning off that gene, by shutting it down, by putting the brakes on, and what I'm showing here is you see this mutant form, the one with the genetic change in a gene called BRAF, which is also mutated in melanoma. You can turn that off, and that X represents cutting down on the growth of the cell. Now, unfortunately, both in melanoma and in lung cancer, instead of having an on-off switch, it's actually more like a dimmer switch. You can only turn it down a bit, and the light goes down a bit. And what I ended up learning in melanoma is that if you turned off something that melanoma activates, a downstream one, by hitting 2 different parts that are activated, you could have an increased effect on the tumor by hitting it in 2 different places. Something that we learned in cancer decades ago, that if you can give drugs that don't have overlapping toxicities 2 drugs are generally better than 1 drug, especially if both are effective. So it was learned that both these drugs are effective. We took those lessons from melanoma, and we began applying them in lung cancer. So when we turned off one, we put the dimmer switch partway down. We went from about 10% of the people shrinking to 1/3. And then when we put the second one on, pushing the dimmer down a bit further, we saw a more dramatic effect with 2/3 of the people shrinking, and now this 1% is being transformed. So by taking the lessons learned both from the systematic genotyping, as well as learning what we've learned in other tumors, we've been able to change the nature of the people. And now it's been 15% plus 20% plus 1% gets us to 21, and we have a lot more work to do to be able to get this to 100%. So with that, I'd like to thank you for the attention.